I'm Daniel Levine, and this is the Bio Report. While there have been a parade of novel therapeutic modalities in recent years, many of them are constrained by delivery challenges. Vesigen Therapeutics is developing novel drug delivery technology known as ARMS that can transport a wide range of therapeutic payloads directly into the cytoplasm of target cells. We spoke to Paulash Mohsen, CEO of Vesigen, about the company's delivery technology, its advantages over viral vectors and liquid nanoparticles, and how it enables the targeting of previously undruggable targets. Paulash, thanks for joining us.
My pleasure, Danny. Nice to be here.
We're going to discuss the area of emerging genetic medicines, their delivery challenges, and Vesigen's platform technology that seeks to address these challenges. It's an exciting time for various gene replacement therapies, RNA therapies, and gene editing, but to what extent is the field constrained by the challenges of delivery today?
Yeah, great question. And just for context, I mean, the past couple of decades have been remarkable for the advances in medicine, right? We went from maybe when you and I were coming up, maybe, treating a symptom or modulating a little bit of chemistry here or there, to now going to absolutely altering the code of life, right? This is truly a remarkable thing. But to get to your question, I think anyone in the field would agree that our ability to perform the edit as an industry has far outpaced our ability to deliver the editing machinery to the locus that matters, right? So if you have an edit you need to perform the lungs or the eyes or any particular organ system, can you get the editing machinery there? Every so often we seem to keep pumping out new ways to edit, but we haven't been able to keep pace on the delivery side. So it's a huge issue.
You're developing platform technology called ARM to carry genetic medicine payloads. Before we talk about the technology and how it works, what problems are you seeking to solve? What advantages might this offer over viral vectors or liquid nanoparticles?
Yeah, and some context from ASGCT, the Cell and Gene Therapy Conference in Timor several weeks ago. That's probably where most everyone in the field descends. And to your point, delivery featured very prominently, and it kind of falls in three categories. The first, as you mentioned, the viral vectors, which are the earliest and probably have the most programs, but they also have very well-known limitations, right? It's a virus, so it has to be one and done. You can't do a second dose because your body's going to come up with an immune reaction. They also have a limitation in the cassette size, so you can add only so much alteration, right? So these are sort of well-understood problems, yet you still look for kind of the next best capsid. The second category, lipid nanoparticles, made very famous probably by the COVID vaccines from Pfizer and Moderna. So these are terrific, right? And sort of earned a place in everyone's knowledge banks. But that was for a vaccine, right? Which is very low dose. Therapeutic doses are, 1,000-2,000x more than vaccines. So you start running into issues of dose limiting tox. And as it turns out, getting to organ systems past the liver has been maybe more of a trick than the industry expected. So some emerging limitations there too. So a lot of the attention at ASGCT was really in that third bucket of non-viral and non-LNP. And there's sort of lots of activity, tons and tons of posters. And we kind of fall in that category. So really, in summary, to answer your questions, we're looking to get past the limitations of viral vectors and kind of the redosability issues. There are some safety issues. Lipid nanoparticles are a foreign product, so you're going to get some sort of an immune reaction at some level. So we want to get kind of past all of these and preserve our ability to deliver editing machinery to very specific organ systems and very specific cell types within those organ systems.
Well, what are ARMS and how do they work?
Yeah, so it's a human transport system. So I'll unpack the acronym for you. That's ARDC1, mediated microvesicles. So ARDC1 is a protein. It's naturally expressed in all human biofluids, and its job is biogenesis of this class of vesicles. So you imagine a cell that's got ARDC1 in it. That ARDC1 is sort of constantly producing and budding from the cell surface these little pouches, little vesicles. And this is used by the body for transport and signaling. And what we do is we sort of hijack, co-opt is I guess a nicer word, co-opt that process for delivery of therapeutic payloads.
This technology was developed at Harvard. It grew out of an effort to understand the way cells communicate. How did this lead to a delivery technology?
Yeah, you're exactly right. It's all about kind of intracellular signaling and transport. And the lab of our scientific co-founder is the one that uncovered ARDC1 and named these arms. So what they also did, and has been developed by the company since, is really see if you can attach your therapeutic payload. So if you have like a gene editing complex, CRISPR-Cas system, or you have a protein to deliver, or you have an RNA, can you take those payloads, attach it to ARDC1, right? And then when ARDC1 is actually creating that vesicle, it literally loads that payload for you inside the lumen or the intravesicular space. So as it's budding from the cell surface, you have these little vesicles, and safely nestled within that natural lipid bilayer is your payload. So we're basically co-opting this process which naturally exists. And if you kind of overexpress ARDC1, you're going to end up with therapeutic levels of the payload you're after.
And to what extent can this be used to target specific tissue and cell types within the body?
Yeah, great question. So we took a page from the early days of AAV and LNP, and we really just started by saying, hey, let's load some fluorescent reporter proteins in these arms through that technique I mentioned, like fusing them to ARDC1, and let's just see where they go. So you start with an IV injection, say, oh, that's interesting. It goes to this particular set of organs and cells. And then you try an intraocular, an intranasal, an intrathecal, right? So you kind of go through all the different routes of administration, and you generate essentially a map, which gives you exactly which organ systems, and you do some more sophisticated analysis using IHC, and you kind of figure out which cell within that organ system you're naturally targeting. So that's kind of where we started, and frankly, the bulk of the work has been there. We have a dozen, let's call it cellular zip codes we can get to. And then we asked ourselves the question, all right, so we can get there, and in some cases kind of exclusively get there, what diseases have their pathologies in those particular zip codes? Because we should be able to address that. And then a very exciting, more recent development is the ability to add surface decorations or modifications to these vesicles, right? So I said it buds from a cell surface. So if you start with your producer cell and you add some sort of a targeting homing device, a targeting moiety on that cell surface, when these vesicles bud, they actually carry those surface decorations with them. So if you can come up with the right surface decoration, a ligand, which will get you to a different organ, you can start to bias that natural tropism, as we call it, or that natural destination. So that's very exciting, right? Because that really opens up the world of possibilities. So you're no longer constrained to where you naturally get. You can actually say, hey, tell me where you want to get, and let's see if we can find that right engager or that right ligand to add to our vesicles to kind of direct traffic to the zip code of interest.
And does this technology have the potential to take an ex vivo therapy and allow it to be delivered as an in vivo therapy?
Yeah, absolutely. In fact, that last little process I mentioned where you add those surface decorations, we've been trying this with a range of cell types. So, for example, hematopoietic stem cells are very interesting, and all the work that CRISPR and Vertex have done to bring Casgevy to the market, that's all ex vivo, right? So you literally have to take blood out, do the modification outside, ship it off to wherever, ship it back, and then reintroduce it to the body. A very fairly complex process. If this could be done in vivo, this would be very exciting. The question is, can you get to HSCs in vivo? And what we've shown in our labs is that you can actually target HSCs. So this actually gives us reason to be pretty excited that you can start to do some of those same modifications, but do it inside the body. And that gives you access not just to sickle, but a whole range of diseases, right?
So drug developers are sometimes constrained by their delivery vehicle in terms of how large a cargo they can put inside it. Are there any restrictions on the size of the genetic cargo here?
Yeah, so we actually think of this as an advantage of the system. We can actually package a fairly significant amount of cargo in there. So just to put some parameters around that, if you take your standard issue CRISPR-Cas9 complex, including the guide RNA, we can fit 20 of those inside every single vesicle, right? So a lot. And then if you think of some of the larger editors like the base editor, adenine, cytidine base editors, or the next generation, kind of the prime editors, we can fit all of those in as well. So now keep in mind, you're actually loading not the DNA, right? You're actually loading the full protein into the vesicle. So we actually are pretty comfortable. This could be a great way to deliver editing machinery to cells of interest.
And what's known about their specificity today?
Right. So most of what is known, we've really kind of discovered through that, brute force analysis, right? Just finding out where they go. And so far what we've said is, like I said, sort of a dozen zip codes we can get to with a lot of specificity. So when we say, we can get to the back of the eye with a intraocular injection, and we can get to, let's say, the retinal pigmented epithelium. Like, we're getting there and precisely there, right? It's not like there's leakage and we're only getting a little bit of there. Sometimes when we see these presentations from companies, they'll highlight all these different organs, but what they may not show on that page is, look, 2% of the injectate is getting there and 98% still going to the liver. So in our case, they go precisely to the place with no leakage.
One of the constraints with certainly viral vectors is concerns about their producing an immune response. Do you see any kind of an immune response with this delivery technology?
Yeah, that's a great question. And while viral vectors have done a lot for us as a society and as therapeutic agents, they do have that important limitation, right? The body's natural defense system is, I see this virus, I'm going to come up with an immune reaction, so next time I see it, I'll be prepared. Since you're using natural human biology here, you don't get that. We don't have any viral components in our system, right? So there is no generation of neutralizing antibodies. We've seen nothing in the way of neutralizing antibody generation. We've dosed very high levels multiple times, and we've done the very specific assays looking for those neutralizing antibodies. And frankly, we haven't seen any in any experiment done over the life of the company.
I suspect that also implies that these therapies could be redosable.
Yeah, absolutely.
Are there any concerns about potential off-target effects?
So this is a good question. One of the FDA's significant concerns is the one you just mentioned, right? So if you're introducing a gene editor, you're introducing your Cas into the body, it does do the edit you requested, and then the question is hanging out in your eye or wherever it is for the life of the patient. Does that introduce kind of off-target editing risk? Peter Marks has been on the stump on this topic for a long time, and with good reason, right? We just don't know, and it's hard to even know where to look for that off-target effect. I have 3 billion genes out there. Look, where are you going to actually look? In our case, it's actually a very different mechanism. It's transient delivery. So you deliver your editor, it performs the edit, and then within 24, 48 hours, the body's natural clearance mechanisms has cleared everything out. So there is actually no persistent expression of the editor. So we think this dramatically minimizes the risk of off-target. Like, we of course have to do the formal analyses, but even as I described there, you can see how different that transient delivery is compared to persistent expression.
And what can be said in terms of comparisons to existing vectors with regards to manufacturability, scalability, and cost?
Yeah, so it's a lot simpler. So I'm actually a manufacturing engineer by background, right? That's what I studied. That was my first job at Pfizer. This is not complex. It's not as straightforward maybe as a small molecule like a pill of Lipitor like I used to make back in the day, but it's not nearly as complex as viral vectors are. There's just a lot fewer steps. In fact, your whole, we divide the world into upstream and downstream. The upstream steps are where you kind of create your stuff, and then the downstream, so separation, purification. There's only one upstream step here because the cell is doing the heavy lift. So you start with the producer cell line, right? You load it with your sequence of interest. The cell does all the heavy lift for you in terms of, loading and exporting those vesicles. So then you have, a mix of cells and a mix of extracellular matrix, which has your vesicles in there. That's the only upstream step. The rest is the downstream processing is pretty straightforward. You're separating by size, you're clarifying and doing a separation by charge. And that's kind of it. Polish, fill, and finish. I mean, there's very few unit operations to contend with. So we think of the complexity as similar to an antibody manufacturing level of complexity.
And how predictably does the cargo end up in the vector? Is this something?
Very, very predictably, right? So one of the things I mentioned in passing at the beginning is you can overexpress ARDC1. So in your producer cell line, you can overexpress ARDC, you get a lot of it. So when the cell is shedding or budding these vesicles, you're getting a lot, right? Because you have a lot of ARDC1. So you're heavily enriching your extracellular matrix for fully loaded vesicles. So as a contrast, if you're familiar with exosomes, and frankly, this is similar to LNPs as well, what you have to do is you have to create an empty vehicle, and then you have a separate manufacturing paradigm to create your payload. And then you have another manufacturing paradigm to kind of put those two things together through some sort of conjugation, electroporation. There's a bunch of different tactics to do this. And that's a pretty lossy step, right? You'll kind of take a big hit to your recovery rate because you'll get some in there and you get a bunch of empties as well. So here, because the cell is doing that work, it's actually highly efficient at doing that. And your extracellular matrix is going to be, our analysis has shown roughly 90% full of loaded vesicle. This is very, very different from the proposition of kind of single digit or very low loading efficiencies.
Vesigen has programs in ophthalmology, immunology, neurology, and hematology, speaks to the broad applicability of the technology. But what's unclear to me is the business model. Do you intend to pursue your own therapies, or do you seek to provide a delivery solution to potential partners?
Yeah, good question. Absolutely a business model of novel therapeutics. You could go down the direction of kind of fee-for-service delivery. That's not been the intention since before I joined the company, and I've continued to uphold that path. We do believe in novel therapeutics. It's something that we can get very excited around. Now, I wouldn't want to constrain the use of these vehicles. So if we pick our lane and we pick one of those areas you mentioned because, small company, limited bandwidth, I wouldn't want to say no one else can use it. We might absolutely consider partnerships or other ways to kind of promote usage of the vesicle. But either way, the end goal is absolutely one of novel therapeutics. And we'd love to be involved in that process.
Well, given that and given the broad applicability of the technology, how do you prioritize what you'll pursue?
Yeah, really good question. And one that's so hard, right? Because we have a bunch of scientists that are A, very capable, but B, kind of very passionate about doing lots of things, right? There's lots of shiny objects to chase. So really what we do is we kind of look across a range of criteria. One, can we do it? Is it a disease population that we think we can kind of get our arms around as a small company? Is it one that's better served by, a partnership? So it's a fairly kind of detailed analysis. We look at all the places we can get. We figure out which diseases we can treat, and we look at that list, and then we prioritize from there and say, look, which ones would we like to do ourselves? Which ones are we likely to take and potentially offer to partners?
And who would be an ideal partner for you, and how would you ideally like to work with partners?
Yeah, so it's a good question and a somewhat complex one, right? So we've talked to a lot of the larger pharmas, and there's a lot of interest there, but they tend to want to get involved when there's a slightly greater level of maturity, right? So you maybe have been in the clinic and you've got some safety data established, et cetera. So those are interesting conversations, and we're down the path of some level of partnership with some of those. There's a separate class of companies that are gene editing companies, right? And those are very different because they're very stuck at delivery versus a big pharma that's doing some of the gene therapy stuff. They're also doing lots and lots of other stuff, right? So it's a small part of their portfolio. For the gene editing companies, this is kind of what they do. So they feel that pinch of delivery in a very real way. So those could be very interesting partnerships as well. Now they're different, right? Because they're smaller, they have fewer resources. So the kind of partnership may look different. But we've actually entertained conversations with both the very largest as well as the gene editing focused companies.
Well, given that, what's known about ARMS from studies you've done to date?
Yeah, so if you think of the process, right, so this is not, it's not like AAVs or LNPs, frankly, where there are some level of product on the market. So there is kind of an established regulatory framework and pathway. This is new. So we'd have to go through the fairly, well-trodden pathway of saying, okay, here's what you see at the bench. Does that look the same as what you see in your first in vivo, your first mouse experiments? Can you show that mid-size, like a rabbit or a mini pig? And can you ultimately show that in a monkey as the closest proxy to a human that's not a human? Right. So if you sort of march through that cascade of experiments, and ideally you'd want to show what we call translation, right? So what you saw in one setting is the same as what you saw in the other setting, plus or minus. And so far we've done that pretty well, actually. So we've gotten to NHP, non-human primate, which is really the gold standard for preclinical data. So we've got NHP data in a couple of different routes of administration. We have some in the eye, we have some systemic. We've shown nice translation of what we call biodistribution, which you get to the relevant cell type. And getting there is step one, but step two is delivering something functional, like delivering a gene editor and showing through sequencing that you've actually performed the edit. And we've shown translation both of biodistribution and a functional delivery in the form of gene editing. So far, so good. And we're pretty excited about the progress. We look forward to kind of a lot more through all the different routes and maybe some of these in vivo HSC and related, but off to a terrific start.
And where are you in terms of developing your own pipeline? Have you identified a lead candidate?
Yeah, so with the data we have, with the NHP data we have, we're pretty close to being able to declare our first program. We have a bunch of data suggesting Stargardt disease, which is a rare inherited retinal disease. That could be a disease of interest. We also have some opportunities in wet AMD, since we can get to the cell type called the RPE, or retinal pigmented epithelium, and we can get there quantitatively and very safely. And that opens up for wet AMD, instead of having to kind of go to the ophthalmologist and get an injection in your eye every 4 weeks. Could you actually contemplate a one and done? That's a pretty interesting thing. As a third opportunity, hemophilia, where again, there's options out there for patients. If you talk to any hemophiliac, they're having to jab themselves every day or every couple of days. And there's a bunch of folks that are restricted from being able to do that. Some of the gene therapies out there have had questions around their durability, right? 5-7 years. Okay, that's good, but what do I do after that? It's AAV based, so I can't take another dose, right? Because you get that immune reaction, right? So there's still lots of room for a one and done or maybe few and done treatment. So we think hemophilia could be a very interesting opportunity as well. So we have a bunch of opportunities, and we continue to uncover more.
Vesigen completed a $28.5 million Series A round in 2020 when it launched. How far will existing funding take you, and what's the plan for raising additional capital?
Yeah, so the point of the Series A was really to get through the NHP data, which we've now accomplished. So we're actually in the middle of a financing round as we speak, and we're very excited, and this is new even since we spoke. We just got a term sheet in, so we're very excited about the prospects of closing this round relatively soon.
Paulash Mohsen, CEO of Vesigen Therapeutics. Paulash, thanks so much for your time today.
You bet, Danny. Thanks for having me on.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.